Welcome to the video on monographs. You might have gone through the monographs from different pharmacopoeias like USP, IP, BP, GP, and some others. This video is regarding the monographs in the pharmacopoeias, the monograph contents, and the information regarding the monographs. For making this video, I have considered USP monograph as an example, but you can relate and you can use this information for the monographs from the other pharmacopoeias as well because their structure is similar. So what is the monograph? Monograph is a official standard published in the pharmacopoeia, and USP monograph is an official standard published in the United States Pharmacopoeia, that is USP. Whenever you want some information, whenever you want to have the quality requirements, that time you go to the monograph. Monographs are there in the pharmacopoeias like USP for the drug substances, for the excipients, raw materials, and the finished formulations. For example, you can have the monograph for metformin hydrochloride API. You can have the monograph for metformin tablets USP, and also you can have the monographs for the materials and raw materials excipients used in different formulations. Monograph is a quality specification for the specific drug substance, dosage form, or formulation, or an excipient. And these standards are the legally enforceable by the FDA in the US for approved medicines. If you are claiming the formulation as USP, it is required to meet the USP standards. Then, structure of the USP monograph. Most of the monographs have this structure that we are going to learn. First is title and definition. So title is the name of the drug like metformin hydrochloride, its official title. It may include chemical name and the description. Identification test. These are also called as ID tests. How to identify that compound or how to identify the formulation. The identification test to confirm the identity of the drug by IR spectroscopy and by the chromatography techniques. Generally, two techniques are used for the identification. First is IR, second is HPLC. These are the generalized identification tests. Then is the assay or strength and content. It defines the acceptable range for the active ingredient. Example, the formulation should contain or the API should contain the active in the range of 95 to 105% of the level claim. If you are working on to a tablet, it will be the label claim, and also if you are using the monograph for API, then it will be the content or assay. Generally it is 90 to 110, sometimes it is 95 to 105, and sometimes it may differ. It is based on the formulation. Then impurities and the related substances, these are also called as the organic impurities. These are the limits for known and unknown impurities, degradation products of the API and the formulation. Then is the dissolution disintegration test. These are the performance tests. These are for the formulation and not for the APIs and excipients. Dissolution disintegration test ensures drug release properly in the body. Other quality tests like uniformity of dosage units as per the USP chapter 905, water content, residual solvents, and microbial limits. Generally, water content are those for those APIs which contain the hydrate forms. Packaging and storage information regarding how to store the product and which type of containers closures are used. What will be the temperature condition, storage condition that will be written in this part. How the product must be stored, like light resistant, in the light resistant container, airtight container, below 25° or below 30°. Then reference standards. USP also provides physical reference standard for the comparison and testing. Example of the monograph is metformin hydrochloride tablets. Definition is the tablet contains metformin hydrochloride. Identification by IR and it matches USP reference standard. Assay, each tablet must contain 95 to 105% of the label amount. Resolution. Example here I have taken like 900 ml of 0.1 normal HCl using paddle apparatus 2 at 50 rpm and the Q point limit is 80% in 30 minutes. This is the hypothetical example I have taken. You can have different resolution requirements and different specification based on the monograph. Then is the impurities. So specified limits for the known impurities and the total impurities might be there. Generally, total impurity limits are not there and sometimes it is given. Then the impurities are also required to be matched and checked with the ICH guidelines. Packaging and storage information, like preserve the tablets in the tight containers. So these are the contents of the monograph. Now why the monographs are important? Why the USP monographs are important? Because these are required to ensure the drug quality, safety and the efficacy. And monographs provide the uniform testing standards worldwide. So if you are working onto a formulation which is to be marketed in the US, that time USP monograph is very important to meet, and it is a legally binding for the manufacturers to meet the quality requirements and specifications as per the USP monographs. Then the monographs are used to make the specifications. Then it supports the regulatory approvals like FDA, ANDA, NDA and others, and it facilitates the generic drug development because if USP monograph is there, you will get the ready reference to meet the quality. So a USP monograph, it is an official document, it is an official standard which is required to meet. Then quality test for the specific drug. It references general chapters for the test methods like resolution chapter 711. It interprets the information based on the information given in the and instructions given or guidelines given in the general notices. So these general notices apply to all the monographs and general chapters apply to the requirements whenever there is given in the monograph and also whenever you are going to develop the in-house method. I have also made a video on USP monograph general chapter and general notices. I request you and suggest you to go through those videos so that you will have the information. I hope this video will help you to understand the monographs, and I will suggest to go through the monographs from different pharmacopoeias to learn more regarding this topic. Thank you for watching the video.